So welcome to the 788th monthly meeting. This is our Norman Willett Memorial Lecture. Before we introduce the speaker, we just want to refresh your memory. We'll hold all the questions to the end. And there are two ways you can ask your questions. Either you go to the participant tab and press this little blue hand to raise your hand, and our moderator will unmute you and you can ask your questions directly to the speaker. Or you can type your questions in the chat box and those questions will be relayed to the speaker. And so we want to remind you that please join ASM. Your support helps us maintain and build this community and network, and it provides a lot of career development as well as the great science. You can go to the membership webpage to update your membership. And right now we have a lot of online material that you can access, keynote speak, lectures, posters, and other scientific programs. And the micro meeting is actually starting now. And so without further ado, the co-chair of our education committee, which is organizing this lecture, Bobby McHale, is going to introduce the speaker.
Thank you, Vincent. Good afternoon and welcome to the 2021 Norman Willett Memorial Lecture. As Vincent said, my name is Bobby McHale and I'm co-chair along with Cynthia Keeler of Delaware Valley University of the Education Committee of the Eastern Pennsylvania branch of the American Society for Microbiology. It's been a long-standing tradition of the branch that one meeting each year would be sponsored by the Education Committee and focus on the many different aspects of microbiology education, from those educating high school students through postgraduate education. This concept was originally suggested many years ago by Dr. Norman Willett, who at that time was co-chair of the Education Committee. Dr. Willett was a professor emeritus of the Temple University Dental School, president of the branch from 1975 to 1977, and a strong advocate for involving students in science education from kindergarten through high school. To this end, Dr. Willett developed science modules that students and teachers could take back to their schools. In order to facilitate these programs, Dr. Willett was the recipient of many grants, including Howard Hughes grants and NIH grants. Dr. Willett passed away in 2014, but his legacy lives on through these annual presentations in his honor. Each year, the Education Committee invites a renowned speaker in the field of microbiology education to share his or her views with branch members and guests. This afternoon, we're delighted to have Amy Chang-Vollmer as our speaker. Dr. Vollmer, by education, is a biochemist and completed postdoctoral research in immunology. Dr. Vollmer has been a faculty member at Swarthmore College since 1989 and served as chair of the biology department for 5 years. At Swarthmore, she teaches courses in microbiology, astrobiology, biotechnology, metabolism, as well as introductory biology courses. Dr. Vollmer's research focuses on bacterial stress response and on the gut microbiome in small animals, and she has supervised the research projects of nearly 100 undergraduate students, many of whom have presented their work at national meetings. She has many papers, book chapters, and reviews to her credit in areas of teaching as well as research. She's committed to science literacy, STEM education, mentoring, networking, and career development. In 2006, Dr. Vollmer was the recipient of the Karski Distinguished Undergraduate Teaching Award from the American Academy of Microbiology. In 2019, Dr. Vollmer shepherded a task force for diversity, equity, and inclusion for the American Society for Microbiology and presented the final report to the ASM board in December 2020. As is obvious, Dr. Vollmer has long been recognized as an expert in the field of microbiology education at many levels. This afternoon, Dr. Vollmer will share her thoughts on microbiology. Nevertheless, we persisted. Please join me in welcoming Amy Chang-Vollmer.
Okay, let me share my screen and we'll get going. Bobby, thank you very much for that introduction. I am really honored to be the Norman Willett Memorial Lecturer because of my relationship with Norm. And so I want to start there. Norm and I met at a 1994 Howard Hughes Medical Institute Program Directors meeting. We saw each other from across the room. He kind of perked up when I said Swarthmore College, and I kind of perked up when I heard Temple University, but we were in a large auditorium and then we weren't seated at dinner. But on the train ride back on Amtrak, I sat down and Norm sat down right next to me. And I think he did most of the talking for the entire train ride until I got off at Wilmington. He asked me all about my career. He asked me whether I was a member of the Eastern Pennsylvania branch. And I said I had joined, but I hadn't come to very many meetings because they were on Monday afternoon and I happened to teach lab on Monday afternoon. And he said, well, you've got to change your teaching schedule so you can come to these meetings. And so many times that I could, I did. And I learned that our Eastern PA branch is really unusual. And I didn't learn this until I became more active at the national level. There are many, many branches all over the country that try to extend what the national organization does to local microbiologists. And every branch is required to have one meeting a year. And many branches only have one meeting a year. I thought all branches were like ours, but in fact, we are quite unusual in the diversity and the inclusion that we represent in our membership and in the different functions that we serve as microbiologists. And I just want to tell people who don't know that among our 10 meetings a year, we focus on many, many different aspects of microbiology. And as an educator, I have found that incredibly useful as I try to keep up to date in my classes for my students. And clearly I'm not a clinical microbiologist or a veterinary microbiologist, but if I can attend a talk and meet someone who's in that field, I can stay updated and refer students to those labs, for example, for summer research. The local branch did support me when I got this idea to create a regional network of microbiologists who teach at the undergraduate level. At a small college like Swarthmore, I'm the only microbiologist on campus. And at many other schools, there are one, maybe a few, and sometimes zero microbiologists. And so I thought it might be awfully lonely. And you have to remember, this was before we knew how to get together on Zoom, that I would reach out to people who'd be willing to drive maybe an hour and a half, who taught at small colleges like, or smaller universities like are listed on this slide. And that we would get together and share some teaching ideas. And then we also got together once a year to have our students present posters. And then we also got together once a year to talk about research. Pictured here in the slide are kind of the people who showed up, the brave people who showed up the first time for my harebrained idea. I want to give credit to Lori Caslake at Lafayette because Lori always organized our research luncheons. I hosted the teaching and poster sessions for 10 years, and I've now turned that over to others. But it's really been a great group. For example, Lori and I, who trained a little bit earlier, didn't know how to do microarrays, and we went to Dave Kushner's lab at Dickinson, and he walked us through that process. We've read each other's grant proposals and manuscripts. There have been people on the list who've written for each other for promotion and tenure. And we started with a mailing list of 14 people willing to drive an hour to an hour and a half. Our radius now extends to like a 2.5 hour drive. That's how much people look forward to getting together. And I'm really hoping that we can do that pretty soon again. So today I want to walk us through kind of the history of microbiology, why it fell out of favor, how it stuck with us, and now it's thriving again, and what we have to look forward to in the future. So in terms of the history, the people on the left side of the slide you've heard of, Leeuwenhoek and Pasteur and Koch, so I won't go into them. I do want to acknowledge Fanny Hess. In fact, I acknowledge Fanny Hess every time I pour agar plates. She was the one, she was married to Walter Hess, who was a collaborator of Robert Koch's. And in Koch's lab, they did all of their bacterial cultures on slices of potato. So you can imagine some things don't grow on potato because they can't hydrolyze starch. And sometimes it's hard to find potatoes. And somebody would have to slice them and keep them moist. And that was just a big pain in the neck. Then they switched to gelatin. But we all know, microbiologists who pour gelatin plates know, sometimes they liquefy again. Some organisms digest gelatin, so they automatically liquefy. And it's a mess trying to keep it of uniform consistency. And so when Walter Hess was mentioning this to his wife, Fanny, she said that she had a friend who had learned from a woman in the West Indies that to make jellies and things like that, that agar was a much better polymer than gelatin. And so literally every time I pour agar plates, I acknowledge her contribution to microbiology. On the right side of the slide, you'll see the three first members of the Delft School of Microbiology, founded in the Netherlands. And Martinus Bjerink is someone who was incredibly prolific. He was studying whatever it was that made tobacco leaves mottled, and he figured out it was something filterable. And at the time, the filters held up bacteria, but let other things through. And so he coined the term virus to describe whatever it was that was making the tobacco leaves sick. He also went on to describe nitrogen fixation, and he described sulfate reduction, a form of anaerobic respiration. Following him at the Delft School was A.J. Cliver. And Cliver discovered that instead of just growing things on agar dishes, if you grew them in submerged cultures, you could harvest metabolites that were secreted. And he found that some bacteria grew much more robustly in submerged cultures. He did a very careful study of biochemistry of different kinds of organisms, and he wrote a book about biochemical unity. He said that elephants and butyric acid bacteria are all the same when it comes to biochemistry. Later on, Jacques Monod talked about what's true for E. coli is true for the elephant without attributing that original idea to Cliver. Following Cliver was Cornelius van Meel, who trained with Cliver and then came to the United States to Stanford's Hopkins Marine Station. And there he described photosynthesis as a light-dependent redox reaction. Redox, by the way, is what makes the world go round. If you're a microbiologist, you know this. If you don't know this, you can't call yourself a microbiologist. He also predicted that water was going to be the electron donor and what was responsible for the generation of oxygen in the atmosphere. He wasn't one to show it, but he was the one to propose that hypothesis. And together with Roger Stanier, he coined the term prokaryotes, which we'll soon learn is now out of favor. For my students who may be in the audience, I want to tell you something. After Van Meel, one of Van Meel's students was Horace Barker, H.A. Barker, who went to Berkeley for his career. He was the discoverer of vitamin B12. And one of Barker's students was Bob Switzer. And I studied with Bob Switzer. So if you studied microbiology with me, we have a direct lineage to the Delft School, and that's pretty cool. Cliver and Van Meel wrote an incredible book called The Microbe's Contribution to Biology that Harvard reissued, and now you can find on Amazon. And Cliver and Van Meel each contributed chapters. You can see Van Meel's chapter on photosynthetic bacteria and on mutations, and Cliver's contributions about metabolism and the flexibility of and adaptability of microbial life. It's a real gem. Cliver also said that his daughter, who had studied general science in high school, had come away thinking that a world with a vague impression of a world teeming with deadly germs, awaiting an opportunity to infect mankind. Probably a lot of people still think that. It seems improbable that this malignant conception of bacteria is generally held. And in reality, civilization owes much to the microbe. Cliver's book was remembered in a wonderful history of biology article by Dr. Singleton and Singleton that came out fairly recently, and it's a great read. So biochemistry and microbiology were partners, thanks to Cliver and others. And through the first part of the 20th century, they elucidated, along with faculty in nutrition, elucidated the vitamins, what they were, what they did, how they contributed to metabolism, all kinds of pathways using radioisotopic tracing, anabolism, catabolism. Some pathways were linear, some were cycles, and some cycles had shunts that were shortcuts through them. In addition, there was an articulation of metabolic interconversion, how you could eat only amino acids and make everything else you needed in the cell, or how you could eat only sugars and some ammonia and make everything else you needed in the cell. The metabolic versatility displayed by bacteria in terms of what you can use for carbon sources, where you can get your electrons and where you can stick them, were all kind of described in these studies in the early part of the 20th century. And of course, bioenergetics had its birth then too. I just remind you about what Cliver said with the elephant. In addition to academic research, microbiology also had huge applications and contributions. You can read a lot about fermentation and how the world wars affected it. As you can imagine, during wartime, a lot of industries had to be switched over, right, to support the war effort. And so oftentimes enemies used to be the source of raw materials, and you obviously couldn't ask your enemy for raw materials. So you had to figure out what you could grow from home. And so one of the things the United States had was plenty of corn byproducts. In fact, penicillin was cultured on corn steep liquor, which is kind of what you get when you take everything that's left over that's not edible and cook it down and make it into a big brown mush. Some students and I, many years ago, bought corn steep liquor from the Sigma Chemical Company and inoculated it with penicillium, and we grew penicillin producing molds, and we showed how it could kill bacteria after about 5 days. But during the war, the military needed to make smokeless gunpowder, and that required acetone. And they also needed to make rubber tires for vehicles, and most of the rubber came from people that they were fighting in Europe. And so butanol is required for rubber. And so along with others, Chaim Weizmann perfected acetone butanol fermentation using the following bacteria. And you can see this Clostridium is named for Bjerink. And yes, this is the same Chaim Weizmann who went on to become president of Israel. So you don't have to be a microbiologist for your whole life. This is a wonderful review about acetone butanol fermentation. And this is the pathway. I've circled acetone and I've circled butanol. If you start with glucose, you can end up at pyruvate. Everybody knows that step. And then this step to the left is what all textbooks jump up and down about making lactic acid so that you can recycle electron carriers. But in fact, really important pathways besides lactic acid fermentation generate more oxidized NAD and substrate level ATP. And the pathway that produces acetone and butanol does both. Obviously, all these solvents are in solution together in your suspension, but distillation allows you to purify these solvents from one another. And that's the fermentation that supported the war effort. Another advance was that you could take a strain. This is an initial penicillin strain, not the one that Fleming found. His grew best on agar. This NR 1951 grew best in submerged cultures so that you could purify the secreted penicillin more easily. But through a series of mutations, so X stands for X-ray, there's some UV in here, there's some N for nitrogen mustard, 10 years worth of mutagenesis. You started with 120 units per mil, and you ended up with over 2,500, 2,600 units per mil. So this is an example of how microbiologists worked to try to increase production of all kinds of products. A more modern table shows the increase of penicillin formation further. And the unit here that you can look at is kilograms of product per 1,000 liter total fermenter capacity per day. All right, and you can see the improvements made at the industrial level. So microbiologists worked on kind of interesting intellectual exercises, but many of them supported lots of industry during the early part of the 20th century. In addition to mutagenesis and being able to screen huge numbers of bacteria for improbable phenotypes, all right, and being able to produce the desired fermentation products and maybe get rid of a few side reactions. All kinds of other tools were used so that people could isolate mutants and study them. I always like to remember Esther Letterberg. She was overshadowed by a more dominant spouse who probably invented mansplaining, but she was the one who investigated many different kinds of fabric and found that the velveteens were best for replica plating. Bacteria could also be grown and selected on differential media as scientists became more facile at understanding the different components that were needed. Temperature sensitive selection was strategic in looking for mutations in essential genes. I've already talked about submerged cultures, and then kind of strain purity and cryopreservation was really perfected during that period of time. And then, of course, the field was changed by molecular biology, what I call round one, right? The physicists who came over to biology wanted to study simple systems. They chose bacteriophage that infected E. coli, and really the phage group went to town, describing many aspects of phage physiology that have really helped us use what they do for our purposes. There was also the transformation. So we knew that we could interact with the bacteria, give them DNA and have a pathogenesis assay. Everybody recognizes this from an intro bio text. And then of course, restriction endonucleases and plasmids. And boy, did that change things. From the archives of microbiology are some wonderful summaries of key discoveries. And I urge you to look at the archives. And each of these discoveries was summarized by a famous microbiologist. And so here's the one on restriction enzymes, and here's the one on plasmids, and using them in vitro for construction. You know, we take for granted drawing things like this, but you can see the years. This was in the late 70s. So just to give you a reference point, I graduated from undergrad in 77. I graduated from high school in 73. So it was a really heady time to be thinking about what you might be able to do. And these are all thought problems, right? I hadn't done any of these. They were just cartoons that I drew with, you know, colored pencils. A little bit later on, or during the same time, people studying viruses were trying to figure out what caused influenza. And the influenza genome was this remarkable fragmented genome. And we recognize today the hemagglutinin and the neuraminidase that can vary in terms of host specificity. So why in the world did microbiology fall out of favor? Well, it was because of molecular biology. It was newer and shinier. And if you could grow E. coli and LB, and E. coli would be your production organism for all kinds of things. And so soon everybody was growing all kinds of things in E. coli and LB. In addition to all the genetic manipulations, people turned their attention to newer, shinier things, eukaryotic systems that were coming online, questions about cancer and immunology. All right, the infectious diseases and other things that bacteria represented seemed kind of dull, and maybe they were going to be solved by antibiotics and vaccines. And so why drive your beat up VW when you can drive something shinier? At this time, there were journalists who were repeating something that they attributed to a US Surgeon General, Dr. William Stewart. But in fact, this paper has exonerated him because after extensive research, they could never find a quote of the exact comments that were attributed to him, that it is time to close the book on infectious diseases and declare the war against pestilence won. But because of the spirit of this kind of talk, research funding shifted to chronic diseases like cancer, heart disease, and muscular and neurological diseases. There was also, Dr. Stewart instituted interesting research into the harmful effects of tobacco. But this urban legend attributed to him was widely repeated by journalists and believed by deans. And so universities started renaming their departments. And the NSF and NIH renamed grant study sections. They took microbiology out of titles and put other things in. In fact, when you were a microbiologist during this time, you had to figure out what study section to send your grant proposals to, because there weren't ones called microbiology. Were you going to take your chance in the cell biology group, where most of the people studied things with nuclei? Or were you going to take your chance with the molecular biology group, and the only thing they knew was E. coli? Microbiology, however, did persist at some ag schools, in vet schools, and actually in industrial engineering schools, where all that industrial fermentation took place. Microbiologists also found homes in departments of biochemistry, genetics, plant path, enology, and viticulture, for example. And microbial chemists were sought not only by the kind of regular industries, but also cosmetic, food and beverage, as well as pharma. So how did microbiology persist when it no longer showed up kind of in the directory of the places that you would normally find it? And it's because scientific questions remain that could be best addressed by studying bacteria and archaea. One of the things that happened was the articulation of the endosymbiosis theory by Margulis and others, but also the kind of evolutionary relatedness of all organisms. After all, the genetic code is pretty universal. People got interested in bioremediation and finding weird bugs that could degrade funny carbon sources. And so people kept studying and kept looking at applications. Microbiology round 2 came around with PCR and the beginning of DNA sequencing, and things like transposon mutagenesis. So they were lifelines for microbiologists. I'm going to take a couple of phrases here and jump on my soapbox. Lots of people teach about mitochondria and chloroplasts. We certainly talk about them in light of the endosymbiosis theory and how that they used to be bacteria. But not enough people talk about the fact that mitochondria and chloroplasts have their own genomes. A DNA polymerase that's much more like the ones from bacteria. They have their own RNA polymerase, again, much more like the RNA polymerase in bacteria. Their ribosomes have 16S, not 18S ribosomal RNA. The ones in the cytoplasm of the eukaryotic hosts are obviously 18S. Mitochondria and chloroplasts have their own tRNAs, and they divide by binary fission. This is something we really don't tell intro students, and I think it's a miss. They're not just these little cute little bags inside the cell. Because they're so important in programmed cell death and enhanced crop production and other things, students really need to know that what makes mitochondria tick and what makes chloroplasts tick. Okay, I'll get off my soapbox. One of the things that biochemists uncovered by and by, was that there's more than way to fix carbon. Yes, there's the carbon-Benson-Basham cycle, right? The one that we know, not the one illustrated here. The one that jumps up and down about Rubisco being the most abundant enzyme on Earth. I posit that it's the most abundant enzyme on Earth because it's one of the most inefficient enzymes. And I remember sitting in an undergraduate biochemistry class when I first saw the Krebs cycle, and I raised my hand bravely and asked, why couldn't we just run the Krebs cycle backwards and fix carbon that way? And the professor looked at me like I had three heads, and I just got laughed out of the classroom. It was really kind of embarrassing. It's like, why would you think that? That just doesn't happen. This is the way we fix carbon. And so years later, when the reverse Krebs cycle was discovered, and that's what's depicted here on the slide, I remember several students in my biochemistry study group from college sending me the paper going, oh my gosh, Amy, you actually were right. You weren't crazy. But there's many different ways to fix carbon. This just happens to be the one that became a chloroplast. And so that's the one most of us study. While I was a first-year grad student at the University of Illinois at Urbana-Champaign, we had these weekly biochemistry seminars. And one Friday at noon, after we had had our clam chowder at the student union, we went into the lecture hall, and there was a man named Carl Woese. Turned out he was on the faculty at UIUC, and he gave a talk. And very few of us got anything out of it, because Dr. Woese, as brilliant as he was, made mumbling and monotone his key characteristics. We had no idea what he was talking about. He mentioned the word archaebacteria. And his slides were not very clear. And so I remember leaving the lecture hall going, oh, well, and I looked over my shoulder. It was one of these lecture halls where the exit was at the top or at the bottom. And so all of us graduate students were filing up out the top. And I looked over my shoulder and I said to the guy next to me, oh my gosh, that must have been really important. Look at the faculty. And all of the faculty were standing down next to Dr. Woese, gesturing and writing on the board. And I completely missed the point. I felt kind of badly about that. And so the next day I walked into a class taught by Ralph Wolf. Now, I was mistaken to think that everybody took a graduate microbiology course like this. But it turns out Ralph Wolf was not only an amazing scientist, but a brilliant teacher. So I got to class early and I sat down and I said, Dr. Wolf, I'm afraid I missed the whole point about Dr. Woese's lecture. What are these archaeobacteria and what do they mean? Dr. Wolf very sweetly picked up a piece of chalk and went to the board. And he said, well, Amy, you know, I grew up on a farm, and the first thing you do when you go get up in the morning is you go milk the cows. And I'm sitting there going, and what does this have to do with my question? But politely, I listened. And he draws the seat and he starts to draw the legs. And he says, the thing about milking a cow is you've got to get a stool that has 3 legs that are the same length, because if you don't, you're going to have a miserable time milking the cow. You'll be spending more time adjusting the stool than milking, and the cow's going to be mad and your dad's going to be mad. And so you look for a stool with 3 legs. Well, by this time I was, like, maybe I shouldn't be in grad school. I didn't understand the lecture yesterday, and I don't understand the milking cow thing. And so then he said, and what Dr. Woese presented yesterday was this leg. The ribosomal RNA leg is very strong, but we need 2 more legs. And he said, and we're working on these things, but we're not quite ready to be as bold as Dr. Woese. One of the things we're working on, he said, is methanogenesis. Taking CO2 and adding enough electrons to it to make methane. And that's chemistry we've never seen before. And in fact, in my lab, we're studying a new coenzyme called methylfuran. And there's a young fella in my lab named John Lay, who is actually a Swarthmore alum, who was my TA for micro, and I was his TA for biochem. We've become really good friends. And he said, and this may lead to some new chemistry. He said, and Amy, there's some people studying lipids. You just wouldn't believe how different the lipids are in these bugs. So here's leg number 2, John Lay's leg. And here's the lipid leg. So here are lipid bilayers that we've seen in both eukaryotes and bacteria. There's some branched fatty acids. You can also see some of these in bacteria. But if you look over here at examples 1 and 2, what you see are not bilayers. These are monolayers. Not only that, the linkages here are not ester linkages that are acid and heat labile. They are ether linkages. Organic chemists have to go to a whole lot of trouble to break an ether bond. And so thermophiles, for example, might be able to hold their membranes together because they're not these bilayers the way they are in the organisms that most of us know. That was our third leg. This allowed microbiologists to jump up and down on this stool and scream, archaea, 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 archaea. Eukaryotic biologists, however, turned a deaf ear to this until the first archaean was sequenced. And the majority of its open reading frames did not resemble anything in any database at the time. And finally, about 15 to 20 years after that, archaea started appearing in introductory textbooks. And now everybody's jumping up and down on the stool. Dr. Woese was not a great spokesperson for this, but he had some disciples, including George Fox and Norm Pace, who were eloquent and active in convincing everyone that this was real. George wrote a wonderful commentary reflecting about his years in the Woese lab and the legacy thereof. And Norm Pace did a lovely retrospective about mapping the tree of life. Norm also had another little soapbox he was jumping on, which was the fact that he didn't think we should use the word prokaryote anymore. That the bacteria and the archaea were so different from one another, it was false to lump them together because they had the absence of something. And so the microbiology textbook I've been using for at least 12 years has, in fact, Joe Willey told me, she said, we did a search and find for prokaryote and replaced that with bacteria and archaea. And we did a search and find for prokaryotic and replaced that with bacterial and archaeal. And so in fact, microbiology textbooks, many of them haven't had the word prokaryote in for a long time. And so if you know any eukaryotic biologists, I would say it's been 15 years plus, it's time to stop saying prokaryote. Here is the tree of life as depicted in Norm Pace's review with unresolved branching highlighted, but we're not done. There's commentary about a fourth domain of life in the study of giant virus genomes. Some viruses being large enough that you can see them with the naked eye. They would not come under the filterable heading that Bejerink came up with many years ago. These viruses have translation machinery that doesn't look like it's related or it could have descended from any of the other domains. Not only that, John Eisen's group has been looking at a lot of the metagenomic data from the Global Ocean Sampling Expedition, and they were looking at REC-A, the homologous recombination enzyme, and RPOB, one of the large subunits of RNA polymerase, along with a translation machinery, and they are talking about stalking a fourth domain. So it's not over yet, folks. Coupled with all of this going on, horizontal gene transfer and the mechanisms that underlie them were really delineated, all right? And the role of viruses in moving pieces of genomes around was really kind of astounding, such that the words fluid and mosaic didn't just describe lipid bilayers, but also were used to describe genomes. All of this obviously was harnessed into biotechnological tools, many of which we use in our labs. Meanwhile, people who weren't looking at medically useful things were looking at other things, extreme phenotypes, extreme physiology, right? How do you isolate an endospore? You take a culture that's sporulating and you boil it, and then you plate what's left, and the only things that will grow were things that used to be spores. Magnetotaxis, so interesting. All of these crazy phenotypes differing from our kind of 37 degrees, slightly salty laboratory growth conditions that were amenable to the cultivation of pathogens. Industry also used bacteria in many ways. There was a huge controversy in California when I was teaching at Mills College in the early to mid 1980s about ice minus bacteria, Pseudomonas, and spraying them on strawberries to keep them from freezing. I remember an Alameda County board meeting where somebody stood up and said, well, I don't want to raise my children in a county that has any bacteria in it. And I just stood up and walked out of the meeting. I decided I just couldn't stay. The discussion wasn't going to go anywhere that I thought would be useful. The Exxon Valdez, the giant oil spill in the Alaskan Sound, utilized the first patented bacteria, oil degrading bacteria. And the Sistine Chapel's artwork that was restored between, I think, 1980 and 1994, used pastes made up of bacteria that were able to reduce the metals that had been oxidized in the paints. And the difference is pretty stunning. So how has microbiology thrived then? Well, microbial ecology to me has been the biggest change in the field. Instead of studying LB pure cultures and log phase shake cultures in the lab, people decided to start looking, at real life. Okay. And there are so many advances in this area, but I want to highlight a couple. The first thing I want to highlight was described by Debbie Siegley and Roberto Coulter as life after log. And in Roberto Coulter's lab, a promising young postdoc named Steve Finkel described this phenomenon called gasping. And I'll show you a figure about that. I met Steve at an ASM meeting. His boss, Roberto, was supposed to lecture, but at the last minute, Steve replaced him. And I walked up to Steve after that talk and I said, Roberto's a pretty good speaker, but you know what? You can go home and tell him you're better than he is. And that was the beginning of a very long friendship that I value very much. Community dynamics, bacteria cooperating, growing in consortia, maybe not being able to grow without one another. So the idea of a single pure culture kind of goes out the window. There's a really nice story about the pink berry bacteria that I'll also be telling you about. The idea that bacteria don't just float around in solution, but form biofilms, form really sticky extracellular matrix that's like waterproof glue, was interesting not only just intellectually, but to companies like 3M who would like to make waterproof glue. The idea that microbes could be beneficial, oh, Cliver would be smiling, and their impact on the environment and the carbon, nitrogen, and sulfur cycles that bacteria are responsible for at least 80% of. Remediating toxic sites, which became a big deal, right? As well as microbes benefiting host immunity and host health, and more about that in a little bit. First, gasping. Gasping is growth advantage in stationary phase. Finkel described, in addition to log phase, stationary phase, and death phase, which is what we teach in a standard microbiology class, there is the 1% of cells that survive can be maintained. Look at this, look at the x-axis. You normally never see anything beyond hours for a microbial growth curve. This is days, months, and in Finkel's lab, years and decades. And in fact, when he looked and tracked different sub-communities, you could actually see succession, okay, that was underlying this overall curve. This really changed the way we thought about bacteria. And really, when you think about it, most bacteria out in the environment aren't living, you know, the high life of LB in a shaking culture, right? It's feast or famine. And oftentimes it's famine. And oftentimes they can persist. Sometimes they persist to a point where when you try to re-inoculate them, they might not grow very quickly or very well. And so the idea of kind of viable but non-culturable also came around at this time. Anyone who's been up to Woods Hole's microbial diversity course, the famous summer course, and gone out to enrich from the environment, probably every summer, at least 1 or 2 students in that course enriches and finds the pink berry bacteria. And one year, Lizzie Wilbanks, one of my former students, did the same thing. But Lizzie wasn't satisfied with just seeing how cool they looked. She wanted to know what's going on. And after a series of studies, she figured out that it was a consortium of the pinkberry 1 and pinkberry 2 subgroups, one of which fixed carbon that the other one used, one of which took reduced sulfur and oxidized it, and the other one returned that sulfur into its reduced form again. And so after decades and decades and decades of students purifying the pinkberry bacteria from this salt marsh that's full of sulfur, Lizzie and her colleagues figured out the chemistry. Don't forget those 6-foot-long tube worms that the submarine Alvin first set its eyes on so many years ago. The question you should ask as a biologist is, what in the world are they eating in a place that has no sunlight? The light here comes from the submarine, right? There are no visible plants to do any carbon fixation. What's going on? When you cut one of these worms open, it doesn't even have a regular digestive tract. So now the question is, how do you get to be a 6-foot-long red worm with no digestive tract? And of course, the answer is you have a symbiont that fixes carbon, okay, in a light independent manner, and that the worm supplies sulfur that drives the energetics of this cycle. So this red tube worm symbiont was the subject of Irene Newton's microbiology paper. So in my microbiology course, I have a set of exercises, some of you have heard this, called Adopt a Bacterium. And every student in the class adopts a different bacterium, and it can't be any of the ones that we study in the lab. And you write short papers about the bacterium, how it got its name, how it was discovered, what its physiology is like, what its genetics are like. And Irene Newton came to me in the class, and she was very honest, and she says, look, I'm taking this class because I have to take a cell and molecular class, and this one seemed less awful than genetics or cell biology. So I'm here, but I have no idea what to study. I'm a marine biologist. And she said that in a way that kind of implied, and of course, bacteria have nothing to do with marine biology. And so I mentioned these red tube worms to her, and she adopted the symbiont that lives in the red tube worm. Irene graduated from Swarthmore, went off to Harvard. She ended up in Colleen Cavanaugh's lab, describing the genome of the clams symbiont, the clams that live right next to these red tube worms. And so she became a microbiology convert. Then there's bacterial evolution. Oh my gosh. Long-term evolution experiment, which unfortunately was interrupted by the pandemic, but I'm sure has restarted, where you can grow bacteria in identical vats, maybe 12 of them. Every 5,000 generations or so, take a sample and freeze it away, and then look for the appearance of new phenotypes. And so the famous one was selecting for a citrate plus E. coli. Many of you who've done a bacterial unknown lab know that one of the hallmarks of E. coli is that it is citrate minus in aerobic conditions. And so they basically selected over many, many generations for a citrate plus E. coli, and then went back and sequenced genomes to figure out which mutations were responsible and whether they had to occur in a certain order or whether there was a really important founding mutation or not. Richard Lenski's lab has been mentoring many, many scientists. One particularly promising young scientist I follow is Vaughn Cooper at Pitt. He has taken bacterial evolution and now studying other kinds of phenotypes, including biofilms. And they're developing genome-based diagnostic for bacterial infections. So this is a really interesting area of pursuit, in my opinion. And then, of course, social media. I'll let this video do the talking.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
Okay, so that came out in 2016, and I would say no fewer than 50 of my former students sent me that link. I think that social media did a lot to show how you can study bacteria and in real time, talk about evolution. Another wonderful contribution in the area of kind of educating the public about microbiology is Elio Schechter's blog, Small Things Considered. And yes, he has been interviewed on All Things Considered. He has this wonderful blog that summarizes really interesting stories in microbiology. And he has been joined by Roberto Coulter. I already mentioned Roberto. He was Steve Finkel's mentor. And I've known Roberto, oh my gosh, probably my entire microbiology career. Roberto's retired from Harvard and has now joined Elio as a co-blogger. Many of my students have Small Things Considered as their homepage because it's so interesting. The other thing that is not as well known is Roberto and Scott Chemleski produced a book, a photographic exploration of the microbial world called Life at the Edge of Sight. It's really stunning. And I have a copy of it. If you're here on campus, you're welcome to borrow it. But I get yourself a copy. It's like a coffee table book. So spreading the word about bacteria, how fascinating they are, how you can answer questions, biological questions using them and live to tell about it, like in terms of evolution, is a really exciting advance. Microbiology thrived because of the people who stayed in the field. They persisted. They didn't change their colors and call themselves some other kind of biologist. They continued to study microbiology, even if they had to be in a department called something else. For me, I had a whole set of communities that allowed me, as the only microbiologist on the Swarthmore campus, to thrive as well. And I will say, oftentimes people don't say this enough, but as a scientist, one of the things that I've really enjoyed is the friendships that I've formed because I've met people through the science. I mentioned Steve Finkel. I have a really wonderful friend named Heidi Kaplan, who she and I have been roommates at this one Gordon conference for a couple of decades. It's a great place to make friends. So the MicroNet I told you about, this group of microbiology educators in the region. National ASM has been a fantastic place for me to meet leaders in the field and for me to contribute to governance and other things. This branch and so many other branches and student chapters that I visited are all keeping microbiology alive, and did so during the period of time where microbiology had pretty much disappeared from many academic institutions. NEMPET is Northeast Microbial Physiology, Ecology, and Taxonomy. It's a group that is coordinated by Craig Phelps, who was actually in the lower right-hand corner of that first picture that I showed you. They meet every June, kind of toward the end of June, up in the Adirondacks. The only bad thing about it is the black flies. Everything else about it is a fantastic meeting. Actually, it's bacterial stress response, but this is a Gordon Conference community that I've been a part of. And also beneficial microbes has been a really great group. In terms of microbiology education, the American Society for Microbiology Conference for undergraduate educators is open to people who teach any kind of biology. And it's a wonderful place to discuss pedagogy, assessment, all kinds of things that educators face. I was fortunate to be the founding editor of what has now become the Journal of Microbiology and Biology Education. It was called Microbiology Education to start with. And I was privileged to lead the Waxman Foundation for Microbiology for its last 10 years. So what does microbiology hold for the future? Oh my goodness, such an exciting time to be a microbiologist, because microbiology departments are back and they're thriving. For one thing, there's more advances in art restoration. Again, this New York Times article was sent to me probably at least 25 times. Okay, send in the bugs. The Michelangelos need cleaning. So the story is in Florence, in the Medici Chapel, because there were fewer people around, the restorers, the restoration people decided to take on a huge project. There was a whole bunch of grime covering some of Michelangelo's sculptures. And the reason is crazy. If you know anything about Italian history, you know about the Medicis. They were quite a colorful family. And one of them in particular was assassinated by his uncle. And they wanted to get rid of the body so quickly and didn't trust any undertaker with it. They just threw it into the tomb on top of other properly prepared bodies. And over the century, Alessandro basically seeped into and all over the sculptures. And so during the pandemic, the restoration folks deployed Serratia Ficaria SH7. SH7 ate Alessandro. which I thought was just so great. Basically cleaned up all the organic residue that was leading to all the staining and deformations. And now the sculptures are once again beautiful and shiny and not grossly contaminated. So that was just an update on art restoration. Round 3 of molecular biology, the result of the incredible high throughput sequencing, sequencing a microbial genome in a day, right? And being able to compare microbial genomes with one another. After all, the human genome sequencing project had to have a lot of model systems also sequenced, or else we wouldn't have any idea what most of the genes encoded and would have no way of testing whether our hypotheses about what they did really rang true. Microbial systems really are the best for this. Those genomic sequences become annotated. In fact, some of my seminar students have participated in annotating genome sequences. RNA-seq, CRISPR now have allowed us to pursue hypotheses that we couldn't even dream of. And one of the things that's happening is that metabolism has come back. When molecular biology showed up, a lot of people stopped teaching metabolism. Would you rather be able to teach about cutting and pasting genes and all that kind of new stuff, or just pathways filled with chemical reactions, right? And so, in fact, more and more papers were talking about the importance of the metabolome and metabolism. And so Elio Schechter actually invited me to write a piece for Small Things Considered, and I named it Hello Again Metabolism, kind of like Hello Dolly. It's good to see you back where you belong. And what's old is new. In fact, this is my VW. So I just drive a bright, shiny new one. Here's an example of how I pivoted. So in my early years at Swarthmore, I taught an advanced microbiology seminar. And the model systems were bacterial model systems. Development, differentiation, interkingdom transfer, all kinds of phenomena that people really don't think microbes do. And through it, my students learned bacterial genetics and a lot of great science. Along with reading papers having to do with these various systems, there was also a laboratory component. And I mentioned before that we learned how to do microarrays thanks to a consortium that allowed us to purchase DNA chips at very low cost and reagents at very low cost. And thanks again to Dave Kushner, who actually walked us through how to do this. And so on the day that my students got their list of genes, right, that were either overexpressed or underexpressed in this comparison condition, there we were staring at this list of genes. And I was so excited. I looked, oh, there's purine metabolism, there's pyrimidine metabolism, there's branched chain amino acids. And my students looked back at me as if I were speaking some other language. I looked at them and they said, what in the world is a branched chain amino acid? What are you talking about? I said, don't you learn these pathways in biochemistry? Purine biosynthesis, pyrimidine biosynthesis, all the amino acids? And they shook their heads, nope. I said, you don't know what any of these enzymes do? They went, nope. I said, so you learn central metabolism, glycolysis, Krebs cycle, yep. Pentose phosphate, what's that? Oh gosh. No intermediate metabolism. Who's teaching pathways? I started to get very upset. When I was exasperated. I sat down and I said, all right, you guys, let's just start with the basics. Where does ribose come from? And as a teacher, you have to be comfortable with silence. And I decided I was not going to speak until somebody answered. And I don't know how many seconds or minutes went by, but finally, one of the students, I think in desperation because they wanted class to end, said, the Sigma Chemical Company? And so I decided that night that the next time I taught that seminar, which was the next year, I would pivot the topics. So since 2003, I have been teaching metabolism because it occurred to me nobody else was doing it. And so we start with central metabolism and we walk through all the major pathways, what the vitamins and cofactors do, how they make and break down fatty acids, how they make and break down peptidoglycan, et cetera, et cetera, et cetera. And so at the end of my seminar, it's been traditional for me to print out a full copy of the Roche Biochemical Pathways poster and award it to my students. And so, you know, this was 2021. So the 8 of them who were on campus showed up to pick up their posters in my new lab. I have one more shot at teaching 12 more students metabolism before I retire in the spring of 2022. But I've decided that one of the things I'm going to do in retirement is teach other people how to teach metabolism in a fun way. The other thing that makes me really excited about microbiology is the gut microbiome and its connection to human health. I'm highlighting papers written by 3 former Swarthmore students because they're such impactful papers. Mike Maywald did his training in Jeff Gordon's lab. And this was the paper where they talked about the 2 main phyla, Firmicutes and Bacteroidetes, and how you needed both of them for a healthy physiology. Renuka Nayak works with Chuck Turnbaugh as a postdoctoral fellow. And they published a paper talking about the importance of personal microbiomes on and the impact of treatment for patients. In particular, pharmacological treatment, but also other kinds of treatment. And that really what she was talking about here was kind of personalized medicine. We're not going to treat you until we figure out what your microbiome profile is, because that will guide our treatment protocol. And Leah Guthrie, who did this work as a graduate student at Einstein, but who's now at Stanford, is studying, she studied in this paper how the human microbiome affects whether colorectal chemotherapy works or not. Fascinating studies. Fascinating studies. I've just talked to Leah, and I've invited her to come to Swarthmore to give a talk this September. And so maybe some of you can come to that. A huge area of interest is the psychobiome, or the gut-brain axis. And this gut-brain axis, if you want to Google something later, Google the vagus nerve, and you'll figure out how the gut and the brain are connected. My introduction to this area started with an alum, Bennett Lorber, who referred me to Sidney Feingold's work. And Sidney Feingold found that the microbiomes of autistic boys, late onset autism patients, and their siblings were very, very different from the microbiomes of children who were not affected. And in particular, the ratio of Firmicutes to Bacteroidetes and the incidence of some very rare Clostridia. These late onset autism cases arose after many rounds of antibiotics for ear infections or sinus infections. And of course, we know that the Clostridia survive that because they can sporulate. This paper talks about the treatment of multiple sclerosis patients and how the outcomes aligned with different types of microbial biota profiles. So linking MS with the microbiome. Many people are aware that MS patients observe very strict dietary protocols, because when they don't, their symptoms flare. And so it may very well be that the gut microbes respond to that diet. In the area of Alzheimer's disease, There was a recent paper that showed that Porphyromonas gingivalis appears in Alzheimer's brains, and a really interesting paper about treatment. This New York Times article was also sent to me by a whole bunch of students. A psychiatrist recommended that his patients eat oysters. And basically one patient said, oh my gosh, the more oysters I eat, the better I feel and my depression goes away. He didn't talk about it in this paper, but the students who sent it to me said, oh my gosh, Amy, it's got to be the gut microbiome again. But my favorite story about gut microbiome is about the lady who could smell Parkinson's. If you haven't heard about this case, you can Google it later. It's remarkable. But this paper has come out explaining that there are volatile biomarkers of Parkinson's and that they can be attributed to fermentation products from the gut microbes. And I love to imagine the use of a breathalyzer to detect volatiles other than alcohol that might lead to a diagnosis, an early diagnosis, because she could smell Parkinson's before her husband came down with it. She just kept telling him he smelled, and he kept shrugging her off. And then he got Parkinson's, and then they went to a support group full of Parkinson's patients and their partners. And as soon as she walked in the room, she went, oh my gosh, that's the smell of Parkinson's. And then they put her in a study. They had 10 people who had Parkinson's and 10 people who didn't, and they had her pick them out. But she made a mistake. She picked out 11. And it turned out one of the controls developed Parkinson's later. So in fact, early detection. Imagine what we could do with a breathalyzer to diagnose gut microbiome fermentation products. Astrobiology is another field. It's very, very interdisciplinary, but there are always microbiologists in it. There's geobiologists, there's chemists, there's engineers, but microbiologists are important, not only because of the study of life on Earth, but because we understand contamination. And so planetary protection is important, protecting the planet we're exploring and protecting our planet when samples come back. For example, the samples that Perseverance is digging up this week. I taught an astrobiology class this past spring for the first time, and I invited several people who are listed here to come and be guest speakers. And so Chris Chiba, who is an alum whom we honored with an honorary degree, is kind of Mr. Europa. And he talked to us about the possibility of sending probes to sample Europa. Clearly, the people who are studying extremophiles are going to be, not the people who study pathogenic bacteria that affect human beings, are going to be the microbiologists who you're going to want to take on this trip, or who are going to be in the lab to analyze the samples. Aaron Goldman, who's now on faculty at Oberlin, studies the evolution of the genetic code and translation itself. Metabolic diversity means that we might be looking at things that eat methane and methanol. These pathways were beautifully worked out by Mary Lidstrom, who was at Caltech and just retired from UW a few years ago. Fabulous chemistry. When you eat methane and methanol, you end up making formaldehyde as an intermediate. And the question was, how do you keep from pickling yourself with formaldehyde? And the answer is, you never have free formaldehyde in the cell. All of these enzymes are in a complex that has a tunnel going through them. And that's where the chemistry happens. Several of the interesting places in our solar system have methane lakes. And so methanotrophs and methylotrophs are probably going to be like organisms that might live there. Always looking for new ways to shuffle electrons around. And that's one of the things that Linda Vu, who just graduated a couple of years ago, is studying in graduate school at the University of Cincinnati. And we're getting better and better at identifying promising exoplanets, particularly ones that are Earth-like. And Eric Jensen gave us a really great lecture about all of those techniques and the likelihood of finding Earth-like planets around small red dwarfs like our sun. Microbiology is a big tent, and underneath that tent, we include fields like immunology. Immunology is a wonderful field, but it doesn't make sense unless you're understanding what the immune system is facing in terms of infection or beneficial organisms. And so microbiology is the natural partner to that. Really exciting immunology research is taking place in innate immunity now. We used to ignore innate immunity because it was kind of boring. It wasn't new and shiny like adaptive immunity, but it turns out innate immunity is really fundamental. Vaccine technology, don't have to describe what's new there. I am going to tell you I'm redoing my slides for that part of my lectures in micro. And there's wonderful advances being made in terms of autoimmunity, transplantation, and allergies. All of this coming because of omic studies. And some of this having to do with beneficial microbes. If you are a student who's looking for a field where you think you could make a huge contribution, I would focus your attention on carbohydrates. Now, when you think about carbohydrates in an introductory biochemistry class, we kind of skip them. We don't really talk too much about them. We talk about how to break them down into glucose, and then we talk about glycolysis. I have flashcards for amino acids. I have flashcards for purines and pyrimidines. I didn't make flashcards for sugars. I'm not sure I could tell you the difference between glucose and mannose right now, okay? And yet, these kinds of papers are telling us that is in fact the sugar moiety. All right, so this is the textbook figure that I use, and it has a fairly nondescript looking carbohydrate chain on the FC region of the antibody. But in fact, this carbohydrate has a very complex composition. And lack of some of these, particularly the fucose, all right, means more severe disease. That's worth studying, all right? But in order to study this, you've got to be a first-class carbohydrate chemist and be able to determine sequences of carbohydrates unequivocally, which is hard. They all look very similar, and then they branch. The other area of microbiology that should go without saying that's really important is virology. Clearly, we're dealing with the zoonosis, with the current coronavirus, and we're going to need to have a lot more collaboration between people who do medical research on humans and people who do medical research in the veterinary area. If you think about it, veterinary physicians, depending upon what kind of patients you treat, you might have a different ecosystem walking in the door, a different set of ecosystems walking in the door during several appointments in your practice. So they're really broadly trained and quite insightful. We're now able to discern molecular mechanisms of entry, replication, and transmission. And in the next slide, I'll show you a video of viral entry. This simply shows you RNA viruses and how diverse they are. And of course, ecology and evolution contribute a great deal to our understanding of viruses. So this is a video produced by Gail McGill, who owns his own company, and illustrates the entry of SARS-CoV-2 into a host cell. Gail does the narrating.
Few viruses have entered public consciousness and stimulated so many detailed images as SARS-CoV-2, the virus responsible for the COVID-19 pandemic. In particular, the spike protein, which gives the coronavirus family its iconic appearance, has received much public attention. It's a natural target for vaccines and antiviral therapies. The spike protein has two parts. S1, a protective cap that recognizes and binds to host cell ACE2 receptors, and underneath it, S2, a membrane fusion machine that pulls the virus and cell membranes together. But to understand the spike is to see it in motion and appreciate the subtleties of its molecular choreography. This flexible protein plays hide and seek with the immune system. It covers itself with a layer of flexible sugars, glycans, that make it harder for antibodies to detect. It also keeps the ACE2 binding domain tucked away, only rarely flipping up to probe for potential interactions. Upon ACE2 binding, the cap portion of the spike protein falls off, exposing the fusion machinery below. Unstable, like a spring-loaded jackknife, the spike is rigged for action. With a protective cap removed, it unfolds, extends towards the host cell membrane, and latches on. Now that the fusion machine bridges the two membranes, one end embedded in the virus and the other end bound to the cell, it refolds on itself, bringing the membranes so close together that they fuse. Once merged, the genome of the virus spills out into the cell, hijacks its protein factories, and produces many more viruses.
I was stranded in Boston, and Gail put me up, and he showed me his studio. At the time, it was many years ago, they were using like Pixar type animation. And so you have scientists doing the animation, and there's nothing like it. I think you'll agree. So I want to thank you for your attention and for honoring me with the Norm Willett Memorial Lecture. It's truly an honor because of the great affection with which we all held Norm and his huge impact on me and my career. I have many people to thank. At Swarthmore, in particular, I want to thank Bennett Lorber, who's also been a terrific supporter. I'm pleased to say that Bennett's agreed to be the last lecture of my micro course this fall. He has a wonderful lecture called All Diseases Are Infectious, and can't wait to hear his updated version of that. Swarthmore has been an amazingly supportive environment in which I have thrived, and that's due in large part to my colleagues in the biology department who always try to make me be the best version of myself. I've been hosted on sabbaticals at DuPont by Bob LaRossa and Tina Van Dyke. Mark Goulian at UPenn was a great host, and Kathy Matthews hosted me at Rice one year. My MicroNet colleagues have been superb, friends and colleagues in the best sense of the word. And also the Waxman Foundation for Microbiology board members have also been incredibly impactful. I've told you about the great community at ASM and CHOMA. The DEI task force members were people who taught me a lot, and I'm pleased now to be a member of the American Academy for Microbiology and working with them to increase inclusion in boards of governors. Bacterial stress response community is where most of my honors examiners have come from and many of my friends in science. And I've been fortunate to be funded by a variety of organizations, and I really feel truly blessed. So I want to thank you for your attention. I'm happy to take questions. I'm also happy to be contacted by email. I think my slides are too big to send, but I think Vincent will have a recording of this, and so you can watch things again and get all those Google things that you want to look up. Oh, I'm so pleased to see people. Hi, everybody. It's really nice to see all of you. Thanks. Meki has said there is going to be a talk at the MBL about the red berries. Oh, that's so exciting. Thanks, Meki. The MBL has a series of lectures, and because of COVID, they've put them on Zoom. You have to subscribe to them, but there were some fabulous lectures last year. I've also heard that they're putting off their big celebration, big birthday celebration, until 2022. So we'll just have to wait to get together in Woods Hole at that time.
Hi, Amy.
Hi, Cynthia.
I don't see any questions in the chat yet or hands raised, so I'll ask a question.
Okay.
You talked in the beginning about being in the biochemistry class and saying, do you have reverse
Oh, yeah.
Krebs cycle? Have you had any students ask you a similar question that you thought that cannot be true, and then it turned out to be true?
Well, you know, I try not to react the way my professor reacted because,
Okay.
you know, I don't think I asked another question for a whole year, right? It takes courage to ask a question, especially if it's kind of harebrained, which mine kind of was. Unless you think about it, if you just to reverse it, you could fix carbon. I mean, the stoichiometry is there, right?
Right.
But I have had students ask questions and I will be very honest with them and say, I never thought about it that way. You know, do you want to explore that with me and let's see if any work's been done? I'm not aware of anything, but I really remembered the way I felt in that classroom, and all my friends laughing at me, by the way, when we went to study group that night. It's like, oh boy, Amy, you really, you know. And so those were the same friends that sent me the papers right when they came out. So I felt kind of vindicated. And so I really do think that we have to be very careful about what we think is, you know, dogma, right? And Swarthmore, for those of you who don't know, is where David Baltimore and Howard Temin trained. We have a plaque in one of our seminar rooms that talks about reverse transcriptase, right? So thinking out of the box, pushing against the traditions is something we try to encourage. And I have to say, sometimes when I sit in study section, I worry about when we criticize people who look like they're doing something really risky. Because in fact, sometimes those leaps are made either by risk or by serendipity, frankly. So I see Steve Finkel in the room. Steve, how old are your cultures now? Oh, I can't. Can you unmute Steve? There we go.
Hi, everybody. The oldest cultures had to be sacrificed after 67 months.
Okay.
They never quite survived the trip from Boston to LA back in 2000.
Even though you tried.
And we're being recorded, so I'm not going to tell you how I transported them from Boston to LA in a pre-9/11 world. Let's just say, if I tried to bring them to LA the way I did back in 2000, you'd be reading about me in the newspaper.
I do remember that when you had a power outage, you went to the lab and shook them by hand. Yes, I do remember that. That's dedication, folks. That's before generator backup. Anyway, and I see Susan Willits on this call. Susan, I hope you know how much all of us appreciated Norm and what a huge impact he had on us, but also he had an incredible impact on the students who came through his class. He was just a zealot, right? And so you were converted in interacting with him in a way that I think few people have that kind of impact. And I was just lucky enough to get to sit next to him on a train for 3 hours. And I can't even imagine what my life would be like if I hadn't met him and joined the Eastern PA branch. So I'm very grateful and I'm very honored. Well, I think the future of microbiology is really bright. I hope that many of you who are listening are either educators or students. I hope we never stop being students. I hope we always continue to be students. There's so much to learn. We have such amazing things in our toolkit now. I didn't say that back at UIUC, I used to purify BAM-H1 for the campus, right? And somebody else purified Echo R1 and somebody else purified Hindi 3. And we compared our enzymes to New England biolabs and ours were much better, much more potent. But over time, right, those things have been eclipsed. But now when we look in our toolbox, it's not just a few restriction enzymes, right? It's computation, it's the ability to grow many more interesting things. It's all kinds of superb microscopy. And so I think it's just a really exciting, and all the organisms that microbes interact with just make it really exciting. So I appreciate everybody being here, and I'm happy to interact with you on, yes, and of course, Crelox and CRISPR-Cas are all bacteriophages, and the people who studied them years and years ago were vindicated in terms of the work that they did. And I think there are many more secrets that bacteria are harboring that are just waiting for us to probe. We just have to look harder. Anyway, thank you again, everyone. And thanks for your attention. And I really appreciate your being here.
Thank you, Amy. Thank you very
Amy.
much.
Take care.
You're welcome. Happy to see everybody here.